Now you got this, right? So that is gel agarose gel electrophoresis. So fragments, right? Fragments everywhere. But how do you know in this gel which fragment is your fragment? That is important, right? Because you want to take it out, right? You want to take your fragment. If we go to, actually, may I ask a question? Why we going to cut the exact fragment, right? Exact fragment of DNA inside the cell. Can't we extract the DNA and amplify that particular gene by PCR? And then, PCR fragment, easy to get a PCR fragment. From any part of the genome, you can get a PCR fragment, right? PCR fragment is a copy of DNA as well, right? PCR fragment is a copy of DNA as well. So imagine, right? We select primers, right? Primers to amplify this insulin gene. One primer, remember PCR, right? Hope you can remember PCR now. So then it will amplify from here to here, right? Out of the whole genome, you can select it and amplify. With now hundreds and thousands of PCR amplified fragments. Still, insulin gene in there. But doing all these hard things, why can't we PCR amplify and clone that and make a recombinant DNA out of one of them? Yeah, but why we don't use that way? Can I think about it and tell me answer? Yes. Are you listening there, do?
Yes, sir.
Hello? Yeah. So we can't take this fragment, right? Because we use Taq polymerase, right? Taq polymerase is another DNA polymerase, right? But the thing is, outside your body, Taq polymerase have no facilities. Actually, it has a facility actually moving forward and moving backward. So we call in vitro. In vitro means outside the cell, right? So it can still, Taq polymerase is not 100%. Taq polymerase has error rate 1 in 1,000. Or 1 in 10,000 probably, right? So because of that error rate, even one nucleotide they mismatch, one nucleotide error. Imagine, because they are the longer than 1,000 kilobases, right? Sorry, 1,000 base pairs. So longer than 1,000 base pairs. How long is that? So maybe this fragment is probably the smallest fragment here, about 40 base pairs or 50 base pairs, right? So maybe fragment somewhere like that, maybe 500 or 1,000 base pairs, right? So there's a big possibility of having one nucleotide is mutated by Taq polymerase. The error rate of Taq polymerase. So therefore, we don't recommend you to amplify PCR fragment. If you put the steps, or I do the PCR amplification, and I clone that in a vector So your examiner won't take it as an answer. You understand? Because of this problem, we not. Some people do that though, practically. They do that when they get large number of colonies. So they get randomly about 50 colonies. Right? After getting 50 colonies, they do some other technique like sequencing to find out if there is any good one. If there is any good one, they do the whole the way. If it is a smaller gene, you can do that. If it is a large gene like insulin gene, is not very small, right? It must be a large one. So the large gene is hard to. So sequencing is a very hard process. Like it's a time-consuming process. So better do this way. Better you run a gel and find out of these small fragments. So this all the ways, right? Imagine this all the ways. The same DNA is same genomic DNA is running in all these, right? So maybe your fragment, let's say somewhere here, because next one, so this sample, so yeah, this is another sample, and then that fragment exactly the same place, right? But how do you know your fragment is in which band? Right? So finding out that fragment is the next step. So how do you call that? What do you call that? So go back to to answer your the answer paper, right? What is their answers? So there's a separation of fragment in the DNA by electrophoresis. Then they say identify of DNA piece of insert using DNA probe. All right. So remember the PCR fragments, right? So the PCR fragments, the PCR fragments, we can't use them for cloning, but we can use to make DNA probes. DNA probe. So what is DNA probe? So you put a label. Every single PCR fragment, you put a label. They are radioactive label. It's like a radioactive phosphate molecule. Right? The phosphate ligand, phosphate branch, but this phosphorus is radioactive phosphorus. So likewise, we join them. All of the PCR fragments, we label them. Likewise. So now it is a probe. Now, so with this probe, that's what your answer says. With this probe, how do you identify exactly that band? They say identify the insert, right? So what do they mean by that? Identify the DNA fragment which consists your insulin gene. That is not an easy thing. They can give it as a 1 point, but actually, how do you do that? They don't tell that. But if you know, you can tell them, and still you get marks. A technique known as Southern blot would do that. What is that? Southern blot. Let me to summarize what is Southern blot, right? So then you can understand exactly how do we do that. Otherwise, you have to memorize, right? find out the DNA fragment with insert, you got to memorize that step. But if you know what is southern blot, it is very easy to remember that. Right? So okay, this is our gel. Right? This is our gel, and we take all the bands as it is. Transfer all the bands into a white. It's like a white nylon cloth, very white nylon cloth, very shiny white. We call nitrocellulose paper. Right? The, the reason why the nitrocellulose paper has attraction or affinity to DNA, they bind the DNA so firmly, and others cannot get rid of the DNA from nitrocellulose paper, because nitrocellulose paper is made to have a lot of plus minus charges in that, inbuilt plus minus charges, right? So these plus minus charges, DNA also have plus minus charges naturally. They attract to each other and firmly bind, right? So how do we do that? How do we transfer a gel to nitrocellulose paper? A small technique called southern blot. So look at, this is a tray, right? So inside the tray you get a buffer. Buffer means a solution which has controllable pH, because that pH DNA is stable, right? In that pH DNA is stable. So we have buffer, and pH is unique pH, as we mentioned, and also it has very salty. It is very salty. So why is salty? Because one way of denature DNA. Denature means temporary denature, right? So breaking the hydrogen bonds and take the DNA backbones. Backbones mean left hand side chain and right hand side chain separate, right? So we saw that in PCR. Remember, in PCR cycle, first thing is heating the DNA to 99 degrees, right? So when this happens, DNA fragments separate to two strands, double strand DNA becomes single strand by actually cleaving all the hydrogen bonds in between them. Right? That can be done for hot water. You can do that, and the salty water also do the same thing. Right? They can separate to DNA. So remember the nitrocellulose paper. So the way we place them, you carefully look at that. The tray is here, and then you get the buffer in the tray, and then you get a kind of a glass plate, right? Two bricks and glass plate on the two bricks, right? And then we have a kind of a large paper towel like thing, right? Large paper towel we put on the glass, and either side of the paper towel touching the solution. So the paper towel actually attract the solution, right, and take it into the gel. Through the gel, there's this buffer has a particular pathway to go.